we have an amazing panel of scientists today. Now, we didn't invite a panel of paleontologists because we really wanted to highlight just the different types of field sciences that are out there. And they're all really different, each with their own set of challenges. And we thought that was really important that we have representatives from these different field sciences come talk to you and share their experiences with you. So I'm going to, if you guys want to come up, and then I'll introduce you. Just come up and sit down. I'll introduce you after you've done that. And it's a little, everyone's very polite. And so they're going to share all their experiences with you. And we are and so we have some local scientists, mostly local. So, but then also we have somebody who agreed to come in from New Jersey. So we're going to start with Dr. Jessica Ware. She's an associate professor of biological sciences at Rutgers University, Newark. She focuses on termite, cockroach, and my entomologist are going to kill me. Odonate evolution. Close enough? Now, just so you know, I'm just going to do very brief introductions because I want them to tell you about their research. And then next we have Dr. Sarah Bagby. She's assistant professor of biology at Case Western Reserve University. She studies microbial evolution in marine environments. Next, we have Dr. Cynthia Bell. She's professor of anthropology at Case Western Reserve University, and she studies human adaptation to high-altitude hypoxia. We have Dr. Arlene Dent. She's physician associate professor in the Division of Pediatric Infectious Diseases and Rheumatology at University Hospital's Rainbow Babies and Children's Hospital. And she has a secondary appointment in the Center for Global Diseases at Case Western Reserve University School of Medicine. She studies how children develop immunity to malaria and the impact of co-infections on global child health. And last and certainly not least, Dr. Yakuta Bhagat. She's a senior environmental scientist at EnviroScience, and she works on water quality and biological monitoring. So, let's start off. And so maybe if we just sort of go down from either end, it doesn't really matter. Tell us a little bit about the science behind your fieldwork, maybe what questions you're currently investigating, and where do you go to investigate these questions?
Do you want to start at your end, or should I start at this end? Go ahead. So as was mentioned, I study insects, and I'm really interested in understanding patterns of insect evolution. And the insects I work on are mostly termites and cockroaches, dragonflies, and damselflies. And I was telling some of the students earlier, it's kind of great because insects are everywhere, so that means you get to go everywhere to go and collect them. So we've collected on every continent except for Antarctica for the insects that we need. The questions that we're asking are different for each of the insect orders that we study. So for dragonflies and damselflies, they're really ancient taxa, and they seem to have first evolved wings. Pterygoda are the winged insects. They seem to have first evolved wings about 400 million years ago. And so when we try and trace the evolution of flight and the evolution of these insects that have their juvenile stages in the water and their adult stages kind of on land and in the sky, in order to do that, we need to collect things from Australia, from Ecuador, from parts of Africa. And you can kind of trace over 400 million years everything that dragonflies and damselflies have done by looking at the physical traits of these dragonflies and damselflies, and also looking at their genomics or their DNA. We do similar things with termites, and termites have kind of a fascinating history because they have social behavior. So they're really just cockroaches now. We consider them to be just cockroaches that are fancier. So they have kings and queens, soldiers and workers. And as you kind of go from continent to continent, the way that termites interact with each other, their diet, and some of the strategies that soldiers use to defend the colony, those vary. And so that's why we do field work, is to try and to figure out questions about that.
So I'm interested in how microbes solve problems. So bacteria and archaea and viruses are everywhere. They certainly outnumber the audience in this room massively. And they have all kinds of different problems depending on where they live, right? So some of them are absolutely flooded with energy, and they have to figure out how to waste enough that they don't blow apart. Some of them are really, really, really energy limited, and they have to figure out the most efficient way to do absolutely everything so that they can survive, right? Some of them live in fast-moving streams, and they have to figure out how to hang on. Some of them live in low-flow environments where they don't get enough inputs of nutrients, right? There are all different kinds of problems. So what I like to do is figure out an environment that seems like it might have an interesting constraint, and then go there and collect some mud or collect some water and figure out who's there, how they're solving the problems, and then how their solutions spread through the environment. So mostly I've done marine research to date. As a postdoc, I spent a lot of time either myself going to the bottom of the ocean or sending submarines down to the bottom of the ocean, which is great but really inconvenient. And so I'm starting to work more in lake systems now that I'm here, and looking at groundwater as well.
I'm Cynthia Bell. I'm a biological anthropologist, and we are interested in why there's so much variation in the biology of living people. And one of the themes I'd like to pick up that Jessica commented on is when we go to the field, we get new questions. We learn things that we would never even think to ask if we didn't do field work. And the field work that I do deals with how people adapt to high altitudes. You've probably heard of Sherpas, right? And you think of them climbing to Everest, and Sherpas and Tibetans, yeah, that's one high altitude population. Another is the population in the Andes. Another is the population in East Africa. And for about a century, all of our information on indigenous populations living at high altitude came from the Andes. And so we had a certain model of this is what highlanders do. And they must have evolved to do this, and this must be the way that people adapt to high altitude. And when I first came to Case Western, one of my collaborators said, "Have you ever thought of working in the Himalayas?" And I thought, yeah, in my dreams. But he actually worked in the Himalayas. So he said, well, I can help you find a convenient field site. It's only an 8-day walk. And I thought I heard him wrong. I said, you mean 8 hours? He says, no, 8 days. And so I thought, yeah, I can walk. I'm sure I can walk. So my mom bought me a tent. I bought myself some boots. And therein lies a career, because in my first field trip to Nepal, where I did in fact walk 8 days in, it took 21 days to get out because the monsoon had started. Well, I had discovered that everything that we knew about high altitude, which was based on the Andean highlanders, didn't occur in Tibetans. And so there was a completely new question. And I've been working on that question now for decades, expanding it to East Africa. And it is, it's a never-ending, always exciting journey. Thanks.
I'm Arlene Dent, and I study how children develop immunity to malaria. Malaria is one of the parasitic diseases that we've essentially co-evolved with. We've co-evolved with the parasite has co-evolved with us, and it's basically in the parasite's best interest not to kill us, right? So what happens is in a malaria endemic area, a child who is born and raised there, if they survive their childhood and they get treatment if they get malaria, they actually can develop immunity such that by the time they're in their early teenage years, they generally don't get malaria again. Well, it's not that they don't get malaria, they have the parasites in them, but they have enough immunity that they're able to keep the parasitemia, the number of parasites down, so that they don't get sick. So that is what I am looking to study. And some of that is understanding what the targets of our immune system is targeting in the parasite. And it's a wonderfully complex parasite that has a mosquito stage, and then it has a liver stage that's completely asymptomatic, no symptoms in us. And then it goes into our blood cells, and it will replicate and persist. And if you survive that, then you can start developing some immunity. And part of this is to try and identify some of the targets that would be good vaccine candidates. And we talk about vaccines, we find a little thing and we can make a vaccine. Well, it turns out that we've co-evolved over millennia, and these are really complex parasites. And they've got way over 5,000 genes that make all these different proteins that are potential targets or maybe useless to our immune system. And so that's what I'm trying to decipher down. What are those targets? What's important? How can we help children develop immunity through vaccines or through other kinds of treatment?
Hello, I'm Yakuta Bhagat, and I work in environmental consulting. But the way I got to working for the company was just a mix of various academic and private sector experiences. My background is in marine and freshwater biology, and through my graduate training, I specialized in fisheries ecology because I loved to go fishing. And I did it when I was growing up as a kid. My dad often took us me and my brother out on the boat to go fishing on the weekends. And I loved it so much that I decided that, okay, well, I have to try and figure out a way to get paid for this. So that took me through my various academic journeys. I worked on research projects involving doing fish community assessments. So we'd go out on the water and set out fyke nets and collect water samples to assess the quality or the habitat ecological integrity of the site that we were evaluating. Some of my projects have involved doing scuba diving to look at assess the zebra mussel and the round goby populations in some of the nearshore areas of the Great Lakes. Through all those various experiences, then I decided that, okay, well, this is really where I'd like to be, in ecological and environmental consulting. So I get a chance to delve into different projects while also exercising and putting in some of my skills and the training that I've acquired through academia. Some of the projects that I currently work on involve water quality and biological monitoring. So some of the questions that we address as scientists in my firm are to do with whether a certain type of human-related or anthropogenic stressor is affecting a water body, and what sort of effect that is having on the biological communities in the water. So we'd go out and we'd sample the fish or the bugs, or we'd collect sediment samples and we would analyze the samples for various chemical parameters or toxicity parameters. And that gives us a better idea and a better understanding of what we're investigating, which is the quality and the condition of the site, whether there are any adverse effects to human health or to aquatic life. So that's it.
Thank you. Also, I think to follow up on Dr. Bhagat's point, I think all of us standing up here and sitting feel incredibly lucky that we get paid to do this. There are times when I come to her, I'm thinking, I can't believe someone's paying me to do this because I love this. So obviously you guys all do very, very different types of field work. Can you sort of give us maybe a sense of what a typical day in the field might be? I know there's no such thing as a typical day. But, as much as you can, just so that our audience has a sense of what you might be doing out in the field.
Well, I work on things that are out at night and things that are out during the day. And I happen to also be a morning person. So my graduate students, I take them with me to the field, and I think they hate it because at 6 o'clock in the morning, I've already gotten up, I had my shower, I had my breakfast, and I'm ready to go to the field. And so sometimes it takes a bit of cajoling to get the team going. But we usually go out and collect for pretty much from when the sun comes up until the sun comes down. We collect as many insects as we can because I try and take the philosophy with them, with the whole team. We think we're only here for a short time. Usually we go for no more than 3 weeks at a time. So we have to get all the insects that we can, because you're not going to find them in New Jersey. So while we're there, we really try to sample intensively. Then we stop and we have some supper, and then we go out to collect all the things that are out at night. And insects, you can actually attract insects to light, because often some insects use the moon to navigate, so you can trick them by hanging up black lights against a white sheet, and they think that they're somehow getting closer and closer to the moon, but really they're just coming to death, coming to you to die. So we do that for several hours until the nighttime, and then usually, 11 o'clock or so, we start feeling like the evening is getting closer and closer to morning, and so then we go and rest. So that's kind of our day. We work morning, noon, and night so that we can maximize our collecting input.
Yeah, so I think that will probably be a common theme. When you're out in the field, you absolutely make the most of your field time because it is very limited. So normal days, goodness. It depends a lot on what sort of operation we're doing on a given day. There are some days that are fairly calm. You're out at sea, you're transiting from one sampling site to another, and so either you're prepping equipment so that you're ready to handle samples when they come in, or you're watching the sonar to make sure that you are where you think you are, or maybe you're looking over some imaging data, maybe you're handling samples that have already come in. The more exciting days are submarine ops days. So when we went to sea with Alvin, the pilots always want to be in the water at 8 in the morning. And the plan is to be down until 4. And so obviously it's a very small submarine, so most people stay on the surface, and they're running support for the sub, right? And so we might be relaying decisions that are being made to the scientists who are down on the sub, or we might be helping them navigate, or we might be just getting everything ready so that we can handle the samples that they collect when they come back up. What I've done more of is using a remotely operated submarine. So it goes down and there's a pilot on the surface who's got, robot arms to control the sub, and we have to sit shift with them in the control van. Because there are no people on that sub, nobody needs to breathe on the sub, and so it can stay down for a really long time. And so we might sit shift in 8-hour shifts for 2 or 3 days. And in principle, you're on shift for 8 hours, but if it's your dive, you're really going to want to be there more than that. And so you're just kind of there. And then when your samples actually get back to the surface, it's very literally all hands on deck. So the systems that we study are often very sensitive to light, and they're very sensitive to air. And so we need to get them safe as quickly as we can. And you just have to keep working on them until you're done, which is how I know exactly how bad feet can smell if you discover, for instance, that there is a hole in the sole of your shoe when you walk across a wet deck, and then you have 36 hours of sample processing to do, because there's really not time to stop. You just have to get the science done, or else there was no point going out at all.
Well, a day for me involves, let's imagine that I am at a study site at 4,000 meters in Tibet. That's about 13,000 feet. And even though I'm usually there in the summertime because I'm a university professor and I can go in the summer, it's, my day usually starts in a village. Usually I'm living in a village. Sometimes I'm living in a nomad camp in a tent. Often I'm in a village, sometimes in a tent, sometimes in a mud hut. And so I'll get up around 5 a.m. and if I'm lucky, I have a thermos of warm water and can wash up. And then I start calibrating the equipment, hoping that that works and that everything is ready. And then other people start getting up and we'll have breakfast. And then usually I've hired someone to help me recruit study participants. And if I'm lucky, people will start coming by around 8, and it might take an hour and a half per person. And if a lot of people come, I'll work from 8 to 8. And as Sarah said, well, and then you've got samples. I've taken blood samples, I might have taken urine samples, mouthwash samples, saliva samples, whatever. And then those all have to be processed. And if it takes you a really long time and it is really freezing cold even in the summertime, and you're working by candlelight, you just do it. And so typically we'll work until maybe, well, hopefully we're done by 8 or 9 at night and can turn in around 10. So in the meantime, in my case, you're interacting, it sounds like, with fellow students. I tend to be interacting with people who often have never seen a foreigner before. And so, and they've never done things like stand on a scale or had their blood pressure taken. So a lot of what I do is also explain what I'm doing and why I'm doing it. And in fact, explaining to Highlanders about why we're interested in how they can live healthy lives and work well at high altitude makes them feel very proud. And so they say, well, of course we can do that. and so I'm glad about that. So they're long days, yeah.
So I work primarily in Kenya at this time, but as part of our group projects, I've worked in Papua New Guinea as well. And I wear a couple different hats. So as a pediatrician, I often walk into the medical community and will spend time in the clinic. And we can't do our work without a team, and without a team that's in our field sites. So we work with Kenyan scientists, and we work with Kenyan clinical officers and Kenyan nurses who we have a rapport with, that they've been part of this study. And so I like to spend time in the clinic with them. One, I get to understand, how the clinic functions, what the limitations are, what life is like. And I think that's actually one of the neat things about medicine, is that I can actually really be a part of these people's lives, in the sense of, like, they're bringing their sick child, I can help with diagnoses, I can help demonstrate good physical exams, things like that make me feel good. And I also try with our research to bring things that help the community. When there are study participants for our studies, we make sure that we pay for transportation so that they always have access to us. We make sure we pay for the medicine so that they, aren't burdened by undue financial constraints. And studies have shown that people that are involved in studies actually do better because they have more access to care. And I like being a part of that. So that's a really important part for me. As part of our studies, sometimes we do go out to the field, to where these people live. And it's wonderful to be able to walk into their homes, to see how they live, if they have access to bed nets, if they have dirt floors, if they have a window, if they have electronics. All those kinds of things helps me gauge what life is like, and so I can better understand. And I thought I'd tell you about a day of mine that I had when I was part of a study in Papua New Guinea. One of the nice things about malaria research is it's generally in places that are warm. So February is generally a good time to go, and I do like that. I will say that malaria actually was here in Cleveland and actually shaped some of the city structure and city planning here in Cleveland. I could go on about that, but I won't right now, but it's actually really cool. But so this was a study that we had in Papua New Guinea, and it was in children, and it was at a school in Mughal. And basically it was looking at healthy children. We're going to treat them for their malaria parasites and then follow them over time to see when they got infected or sick again, and what their immune responses and parameters were that were associated with that. So the day started pretty early, probably up by 6, in the cars by 7, because it's a good hour and a half, 2 hours to Mughal, this school area. It's a fairly rural area. It was rainy, it was muddy, we did get stuck, you know, that kind of stuff. So with our team of Papua New Guineans, other scientists, myself, and a couple other scientists actually from Australia, and I was the American, we had already talked to the schools, their teachers. We had brought crayons and gifts for people. And so the children are lining up. We've gotten parental permission. We've gotten community permission. All of that takes time and talking and education, which is good. And then, so I put on my pediatrician hat, and we did, you know, basic vital signs. I did a physical exam. Some of these kids had very large spleens. Some of them had very low hemoglobin. Some of them had big bellies. I saw some diseases I'd never seen before, like yaws, which is like a syphilis-related disease. Wouldn't have never seen it if I hadn't seen this kid that, oh wait, we pulled off the soccer field to look at. Then we drew blood, measured heights and weights, gave them medicines, gave them a treat, like a little soda and some crackers to make sure that they were tolerating their meds okay. Took the blood samples. We made slides too, because that's how you see if they're malaria parasites. Drove 2 hours back to the lab, and then I put my other hat on, the scientist hat, and got to processing the blood and doing immune things. And when the generator was working, that was great. And sometimes it didn't. And so you stopped and you put your headlamp on and you could do a couple little things. And then the generator would kick back on. But the windows were open. It's hot. And so the moths are coming into the hood, and you're just trying to manipulate around the moths and just get everything as safe as possible and into the incubator so that everything is safe and you can do a readout. And then by headlamp and propane stove, we would cook, you know, beans and franks and things like that at about 9. And if the rats weren't getting in, we could fall asleep.
That sounds like a typical field day. I'm not kidding either.
No, I'm not kidding either.
All right, well, so a typical day in the field for me, and this is actually, we'll touch upon what some of my other fellow panelists have mentioned, that it does for me as well. Unfortunately, it starts at the crack of dawn. Because like Jessica mentioned, you know, you try to get the most out of your field day, and you want to sample as many sites and collect as much data as you can, and you have a limited amount of time, so you do need to start very early in the morning. Now, it has been a while since I've had some very intensive field work, so I'll go back to my graduate student days, where a typical day in the field would start out in our motel room. We'd wake up at 5 in the morning, and we'd get all our instrumentation and calibrate all our meters, our YSI water quality meters, based on the pH and dissolved oxygen standard measurements that we needed to use. We'd load up our field truck and check to make sure that the boat was secured properly to the trailer. Make sure the boat plug was in. That's very important. I've learned that the hard way, that before you launch the boat onto the water, make sure the boat plug is in. When we get to the site, and this happened to me quite a few times with my luck, especially if it was just me and a couple of other of my female colleagues. Sure enough, we'd get out of the field truck and be on the boat ramp, and there'd be a group of fishermen that were either also launching their boat or were just getting off the water. And they'd stop dead in their tracks, stop what they were doing, and just stand there and stare. Be like, oh, there are some women there that are trying to launch a boat. Let's see how they do. So So as interesting as it was to be under that type of microscope, that's, we had to, as professional field scientists, we just had to carry on and be like, all right, well, if they're going to watch, let them watch. And we won't let that deter us from what we're doing. So we got our equipment together, put on our waders, get on the boat, and we'd get to our first site. And there typically we'd start off by collecting our water samples. We'd set out our fight nets to sample the fish communities there. We'd take sediment samples, dole them out into our little containers that we had in our boat, and that in itself, collecting all those various types of samples, doing all the readings that we had to do, and everything would constitute a good 14, 15-hour day in the field. We'd come back to our motel rooms exhausted completely, and I still remember this very vividly, that while we were still out on the boat and we had about maybe an hour or so of daylight to go, all I could think about was that cup of ramen noodles that was sitting in my motel room that I was so looking forward to. So that's how my typical field day would end.
So all of these experiences, I mean, you guys are talking about these experiences from sort of this lens of having been through it for years, right? So now that you're here and looking at these years and years and years of experience, if you could have written a letter to your younger self that, before your first field experience, what would you have said? You don't have to tell us a whole letter, just like what are the main points you really want to get across? Any takers?
I'll go.
Okay.
Well, so if I could have written a letter to myself, let's say before my very first intensive field season, I think I would have noted 2 points in particular. One, to be patient with myself, and not take things too seriously, but not be too hard on myself either about the field work that I was doing. I think as I wonder if that's something to do with just being a woman scientist and knowing that, especially if you're in a field that is predominantly, or is dominated by a lot of men. There is a part, I think, of us that feel that we need to also show that we're equally capable and we can also do the work. So at times I found myself like putting that pressure on myself and thinking that, okay, well, I need to show that I can do this too. So that's one thing that I would have written to myself, is that just be yourself and be patient. And the second thing I would have written to myself would be have fun. Don't savor what you're doing. And I especially think back to that very first intense field work or field week that I had as a graduate student. We had basically our routine involved being out in the field for 10 days and then getting 5 days off. And well, when I say off, I meant like going in the lab and analyzing the samples for 10 hours a day, and then going back out in the field for another 10 days, working 14, 15 hours a day. And we'd start out the day knowing that we had a certain number of sites to cover and a certain number of samples to collect and all this data to gather. And so right from the start of the day, it would be go, go, go. You have to do this, you have to do this, you have to do that. But oftentimes, I would think back at the end of the day that, I saw this really cool fish. And I caught this really neat thing, and I didn't even get a chance to examine it or look at it and kind of savor the moment or kind of study it a little bit. And I would have done that if I was out fishing as my pastime or my hobby, but I didn't have that luxury of having the time to really relish and enjoy what I was doing and kind of take in that moment, kind of do the figurative smelling of the roses, so to speak. So I think that's one thing that I would have said to my myself that, well, just take some time to really enjoy what you're doing and try and remember why you're there or what got you there in the first place.
Who's next?
Well, I think if I was to give a letter to myself before my first trip, the first time I went anywhere for the field was I went for 3 months to Costa Rica, and I lived in a really remote area where there was only 2 other humans there. We were in the jungle, but we would occasionally, scientists would come to this station that we were at, and I second-guessed myself. And if I was going to write a letter to myself, I would say, don't second-guess yourself. Just be brave, and if you think you don't know the answer, or you think you have a good idea, just do it. And when I look back on those 3 months, since it was my first time in the field, and since I was the only woman that was at the station, and I was the only black person at the station, I guess I just assumed that I would be wrong, and I missed a couple opportunities for some cool data collection that later on I found, or I saw, when I saw it in Ecuador years and years later, man, that was like, geez, I was right on that one, and I should have told that guy to let me finish my idea. So I would have told myself to be more brave and just keep going.
Well, I think I would agree with Yakuta that I would probably tell myself to enjoy where I am and the people that I'm with. As I mentioned, I'm in extraordinary places, on the Tibetan Plateau, on the East African Plateau, and in the course of collecting data, I'm talking to lots and lots of different people. And I'm usually, and certainly when I first started, how old are you? Where do you come from? And I was very businesslike and so forth. And over time, I became more comfortable, and I felt it was okay to just kind of ask people things about the community and say how beautiful it was, and that I felt more comfortable just interacting with people, even though it was part of the research project, as conversationally as people. And it doesn't take that long. And it makes things, it makes me appreciate them and the whole experience more. And I think they also have a sense, as I mentioned, that usually they haven't seen foreigners before. And so, they learn that foreigners are people too.
I think one of the things that I wish I had known is that it's incredibly helpful to know what you need and to ask for it. Because often, I think women particularly are accultured to not take up space and to not make difficulties for other people in getting the things that we need. And it took a few cruises before I realized that the ship's crew is there to support the work of the scientists, right? That is their job. They are proud of their job. And if you don't ask them to do their job, what are you doing? And so it really helps to have that going in as sort of like, right, I need to have a good answer to what do I need to make this work, right? That helps the work in itself, and it also helps the interaction. The other thing is, again, the ship's crew have certainly seen scientists before. They sail with scientists all the time, but they're really interesting people, and it's interesting to hear where they've been and what they've been up to and what science they've helped support. And it helps when you're out. It hasn't been as extreme as 3 people for 3 months, certainly, but there are social dynamics on a ship, right? It's a small group of people, and you're out at sea for a while, and it really helps the work of the ship if you relate to each other as people. And so, again, just talking to people makes a huge difference.
I'll just be really fast. I probably wouldn't have listened to myself if I wrote myself a letter, because I knew everything. But I will say, I wish I knew not to be in such a hurry, because I was always hurry, 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 which is, I think, sort of what you were saying. But I wish I knew to slow down, and I didn't know that for a long time.
Yes, it's all very common themes, isn't it? The things that we learn after years and years and years spent in the field, and you look back and you go, why did I not do this earlier? But I think sort of at the core of it, maybe it actually has to do with like our own, certainly for me, my own confidence like in myself, and that I do know what I'm talking about, and that and I'm trusting my own judgment. And so all of those other things that I used to do all stem from the fact that I probably didn't really trust myself very much because I didn't think that I was good and didn't have the experience.